All right, thank you, everyone. It's a pleasure for me to introduce today's speaker, Dr. Jaime Castrellon, an assistant professor of psychology at UCLA and director of the Castrellon Lab. Jaime is not only a brilliant scientist, but also a friend and collaborator. So I'm especially delighted to welcome him here to CVL's Science Luncheon series. Dr. Castrellon's research sits at the intersection of social and affective neuroscience, behavioral economics, and computational modeling. His work examines how people make decisions that impact their own well-being and that of others, with a particular emphasis on motivation, reward, and social cognition. He earned his PhD in psychology and neuroscience from Duke University, where we overlapped in Dr. Greg Salmon-Larkin's lab, and he completed his postdoctoral training at the University of Pennsylvania, where he deepened his work on the neural mechanisms of decision-making. What sets Jaime apart is not only his intellectual curiosity and scientific rigor, but also his incredible drive and warmth. He brings thoughtful, collaborative spirit to every project, and his work continues to push the boundaries of how we understand human behavior across contexts, across individuals, and across the lifespan. Please join me in giving a warm welcome to Dr. Jaime Castrellon.
Thank you so much for that introduction, Kendra. It's I think really special and unique to be able to be introduced by someone who you've worked with, someone who you've known for quite a long time. I think we started collaborating back in sometime 2014. And so it's nice to join you here. Thank you for inviting me. So today I'm going to tell you about some studies that I've led over the past few years that focus on the neural mechanisms of decision-making and preview some ongoing and future directions in this area. So now this.
Okay.
Oh, there it goes. Okay. All right. So my lab opened at UCLA in July 2024, and our team is really interested in 4 main areas of research. These include understanding the basic mechanisms of motivation and value, decision making across adulthood, as well as social and affective experiences in daily life, and somewhat an extension of that, understanding legal decision making. So today I'm going to focus on this first area, focusing on these basic mechanisms. So I'll provide you with some background, walk you through some of those basic mechanisms, and shift over to thinking about how neurotransmitters in a basics dimension might be extended to understanding social context. And then I'll close out with some future directions and lead you to where our lab is taking this now. So just as you're sitting here thinking, at some point in your life, you might have been faced with a decision where you have to consider whether it be worth it to wait and save your money for that all-inclusive resort vacation, or spend what you have now for a staycation in your city. And this, for me, is Los Angeles. So oftentimes we'd rather get away. Or maybe you had to decide whether to put in more effort and work late on a project to meet a submission deadline, or take your time and focus your energy elsewhere and preserve your abilities. Maybe you found yourself driving along the highway in the city and you see somebody signaling for help for their car. Do you put in that time, effort, and even uncertainty about this person who you don't know to pull over and help them? Or do you drive past, leaving them to go it alone? While these decisions in each of these scenarios vary in many ways, they all involve weighing benefits and costs and are shaped by how sensitive we are to the differences in the subjective value of the options at hand and how motivated we are to pursue them. And so when we think about this sort of motivation, there are a number of key brain regions that come to mind. So for instance, the ventral medial prefrontal cortex, the amygdala, ventral striatum, and the midbrain are all involved in processing reward value and guiding our choices. And notably, some of these regions also receive projections from dopamine neurons in the midbrain that modulate ongoing signals during choices. Now, while dopamine has been studied in non-social decisions, there are some inconsistencies at timing. And dopamine's role in social decision-making is less well understood. So perhaps dopamine plays a special role in shaping motivation when decisions are made in a social context. So crucially, this one neurotransmitter might play a key role in our daily lives, from how we choose to spend our limited time with close to others, or pursuing goals in effortful contexts, or how we decide to help others when we're in a position of power, and maybe even how we correct and update prejudiced beliefs. So I'll try to touch on as many of these today. So our primary questions include, first, how does dopamine support motivation or decision-making generally? And then, by extension, how do these motivational mechanisms shape social context? So let's dive in. To begin thinking about the role of dopamine and motivation, it helps to start with basic causal mechanisms. And so here I begin by meta-analyzing the effects of various dopaminergic drugs on reward discounting in rats. And if you're not familiar with it, discounting is this tendency that we have to devalue rewards that are relatively delayed or uncertain or more costly than sooner, more certain, or less costly rewards. And so what do I mean by this tendency to devalue rewards? So in this figure here, the y-axis represents how much someone subjectively values some reward. And on the x-axis is some kind of cost or barrier that you have to overcome. And with little to no obstacles, a patient person and an impulsive person might value rewards the same. But over time, as this cost increases, in this case time, we see a pattern emerge that this impatient person devalues or discounts this reward more steeply here in red compared to someone who might be more patient and is doing this less steeply. Early work had documented this effect in pigeons, and they found that these pigeons could devalue or discount this in a systematic way that was hyperbolically shaped. This work had been extended onward in rodents and in humans. And so this is something that is preserved across mammal species and non-mammal species as well. While a number of pharmacological studies have been done to tease apart some of the effects of dopamine and motivation, they have been widely variable. So to make sense of the mechanism, I led a meta-analysis of dopamine drug action on reward discounting in healthy rats. And the focus on healthy rats allows us to observe patterns that are not confounded by pathology. So here, we look at discounting of time, probability, and effort costs. And we're specifically focusing on drug actions that tap into first the D1 receptor system of dopamine, as well as the D2 receptor system, and these dopamine transporters that are responsible for reuptake the leftover endogenous dopamine in the synapse. And so the hypotheses that I had for this were guided by a classic model of striatal dopamine and its influence on motivated behavior. So according to this account, these dopamine D1 receptors are part of a direct pathway and have been shown to promote motor behavior and goal-directed outputs. And these D2 receptors are part of this indirect pathway and are shown to inhibit motor output and goal-directed behavior. So given this model, I hypothesized that facilitating D1 receptors and blocking D2 receptors would increase discounting of rewards, and that conversely, blocking the D1 receptors or promoting the D2 receptors would decrease the discounting. So we have a mechanism that we can test, and so what we want to do is evaluate that. What I'm going to show you here are the effect sizes from 5 independent meta-analyses that we ran, one for each type of drug action. And this summary is going to allow us to visually compare the effects. So the effect sizes that appear further to the left are indicating that the drug resulted in lower discounting compared to placebo, and the effect sizes further to the right indicate that the effect sizes resulted in higher discount relative to placebo. And so the first thing that I want to direct your attention to is that there isn't a clear distinction between the modulation of D1 and D2 receptors. So specifically, both D1 and D2 agonism generally have no effect across these studies on discounted behavior. And antagonism of both D1 and D2 receptors both increase discounting in these healthy rats. And then finally, we actually see that dopamine transporters reduce discounting, which is actually consistent with its therapeutic effect in treating impulsivity and ADHD symptoms. So we're starting to see a picture that doesn't really align with our initial hypothesis. And it actually does align with emerging views, however. There's an updated model of the striatum, which suggests that dopamine can be modulated through the ventral striatum to this ventral striatal palatal pathway. And what's special about this ventral striatal palatal pathway is that these D1 and D2 receptors have a shared role in carrying signals in both the direct and the indirect pathway. So this is as opposed to the dorsal striatum, where those differences may still emerge. And so if we think that this reward discounting behavior is more strongly modulated by these ventral striatal signals in the striatum, then we might have some reason to explain why these D1 and D2 receptors aren't easily dissociable. This could also suggest that the relative balance between D1 and D2 receptors is more important than the number or signaling capacity of either one receptor type alone. And so the absence of an effective dopamine agonism could also arise from a well-characterized phenomenon whereby dopamine levels exhibit an inverted U-shaped association with behavior. So the rats in this meta-analysis were all healthy. And so if they have optimal levels of dopamine, then perhaps they've sort of hit their ceiling, and further augmentation by the drugs might not be able to push their behavior so much. So there's probably a limit to the number of receptors, for example. So these drugs won't increase their number of receptors, but maybe we can study natural variation in a sample. So here, our understanding can benefit from an individual differences approach, or we can understand how people vary between each other, or perhaps how clinical status might modulate these effects. And that's what I'm going to show you next. So just some caveats to keep in mind here when we're going to transition to thinking about human studies is that the rats have to be trained on these tasks. So it's difficult to distinguish discounting in humans from the learning effects that happen when we have to run these studies in rats. Humans also rely on additional cognitive mechanisms like episodic thinking and simulation, or much more complex behavior. And human rewards, like money, are conditioned reinforcers versus the primary food rewards that are given to rats. So exploring primary rewards in humans might allow us to better evaluate these comparisons, which we'll also get to later on to the second part of this talk. So our main takeaway here, that we have some evidence for a shared role between these D1 and D2 receptors in reward discounting. So while that first study helped us elucidate those basic mechanisms in rats, we next want to translate this into humans. And so to do this, we combined behavioral testing with positron emission tomography to image the dopamine receptors in humans and identify individual difference associations between D2 receptors and discounting of time delays, physical effort, and uncertainty. So before I jump into that study, I just want to provide a quick little reminder of how PET works, since it's less commonly used than fMRI. And so here what we're doing is we are injecting a radio tracer into participants that binds to a substrate of interest. So the substrate of interest in our work is the D2 receptor, and the tracer that we use is known as Fowler. And so over the course of a few minutes to hours, the tracer is going to make its way to that substrate in the brain. It's going to decay and emit a positively charged electron that's going to collide with a positively charged electron is going to collide with electrons in nearby tissue. And this collision is going to trigger a gamma photon emission, which we can then visualize with this ring of gamma channels. And from that, we can actually construct an image of the radiation and identify where that radioactivity originated from. So this is just an image over time of the tracer in one of our participants. So early on, the tracer is really making its way in, trying to bind to the substrate of interest. And then later on, you can see where it saturates. This is an axial view. So we're looking at a slice of the brain where you can see the striatum that has the caudate as well as the putamen there. And so that's where all of our dopamine receptors are. And that's why over time, the radioactivity signal is really building there. Another way to visualize this is using a time activity curve where the x-axis is time and the y-axis is radioactivity. So this red line represents the radioactivity in a region of interest, like the striatum, where we know there's receptors, compared to this yellow line here, which is representing the radioactivity in a region where we know that the receptors are in little supply, like the cerebellum. And our measure that we're going to be using is called binding potential, which just reflects this ratio of the radioactivity in a region of interest versus this sort of reference region in the cerebellum. That's what I'm going to be referring to later as this measure of that difference. So PET imaging can really provide a fine-grained measure for individual differences, which lends itself well to this approach. And importantly, in addition to just looking across individuals who are healthy, we wanted to study how people might vary in clinical status. So this is a comparison that I put together here, sort of a meta-analysis, looking at past meta-analyses of studies that have used PET imaging to visualize different aspects of the dopamine system in populations with different psychopathologies. So we can compare people with schizophrenia, drug use, anxiety, major depressive disorder, and see how there's variation in these different targets that are visualized with PET. So we can already see, just the main takeaway from here is that there's lots of variation. Because of this variation, we have good reason to suspect that if we compare healthy people to clinical populations, they might look different. So that's what we're going to use. So to bring us back to our questions at hand, I wanted to evaluate natural variation in receptor levels. So specifically, we hypothesized that if the D2 receptors are involved in regulating motivation, people with more D2 receptors might discount rewards less steeply. And so to do this, we collect PET data. This was collected both at Vanderbilt and at Yale. So we have two different tracers that provide complementary coverage across participants in the whole brain as well as the striatum. And so in one visit, they underwent a PET scan, and on a separate visit, they completed these discounting. So they did it for time delays, uncertainty, and effort. These decisions were incentive compatible. So at the end of these tasks, they were told that they would receive an actual payout to make sure that these people were really motivated to actually want to perform. And then following that analysis, we compared our effects to past studies that were done in clinical groups. So I'm first going to show you the correlation scatter plot between receptor availability and discounting for the ventral striatum for probability, time, and effort. The only reason I'm showing you this one plot is because the effects look the same for every region of interest. And that's just to say that there were no statistically significant associations. There were no correlations in these healthy adults for any of our regions of interest, which included the striatum, the hippocampus, the anterior cingulate cortex, the thalamus, the midbrain, missing one. We even did a whole brain voxel-wise approach. No voxels derive corrections for that. So we're not seeing any associations here. Doesn't seem to be a case that the behavior is related to the receptor levels. So how does it look like when we compare our sample, though, to past studies? So as before, I'm going to show you sort of meta-analysis plot and comparing this healthy population to other clinical groups. And so effect sizes further to the right indicate that there is a positive correlation between a dopaminergic measure with PET and discounting, and effect sizes to the left indicate that there was a negative correlation between a PET measure of dopamine and discounting. And so I've seeded our effect sizes right here, just including the studies where we had clear overlap in one of our brain regions in the striatum, and compared these healthy controls, past studies where people have looked at this correlation, to other samples where there's, for example, gambling or alcoholism, methamphetamine use, or ADHD and obesity, Parkinson's disease. And what we see is that the association seems to vary depending on this sort of clinical status. So whereas overall, it looked like healthy controls don't really show an effect. Some groups show a negative correlation, some groups show a positive correlation. It suggests to us that this characteristic in dopamine signaling might depend on some other like baseline characteristics. So we find some evidence first that supported a shared role between these D1 and D2 receptors. But here we find that healthy and clinical populations are showing some different associations. Now, much later after publishing those results, I went back to the data and thought about another possibility. So perhaps dopamine could affect discounting by modulating the sensitivity to the value of the options at hand rather than people's overall preferences. So what do I mean by that? These are different kinds of participants that we simulate, A, B, and C. And so we simulated participants using logistic regression to examine how choices related to the difference in value between options. And so all of these participants behave in the same way in that they chose the larger option 50% of the time. But some subjects are less sensitive to the subjective value of the options, and some are more sensitive. So for example, subject A is not really sensitive. Subject B is really, really sensitive to their choices, to the subjective value difference in the options. Subject C is somewhere in the middle. So people could realistically choose a delayed or an uncertain or a high effort option with the same frequency, but might be sensitive in different ways to the values of the choices at the trial level. We do that in our data set, we actually find that's the case. At least with the ventral striatum, the participants who have more D2 receptors and the BS there are more sensitive to the subjective value difference between the delayed and the immediate reward. So this is just showing you a logistic regression, and what we're looking at is a difference in these slopes here between people who have higher D2 receptor levels versus average and lower D2 receptor.
Sorry, how are you quantifying subjective value?
So this is coming from a computational model where we fit a hyperbolic function to the choices that people made, and using the K parameter of this fit, we can estimate the subjective value at the trial level. Then we put the trial level subjective value differences, estimating that in relation to their binary choice.
So it's basically driven by the low data.
Yes.
Rather than the
Correct. Right. That's right. We next wanted to test whether or not these associations between D2 receptors and discounting might be specifically related to some kind of underlying computation of that subjective value. So if the subjective value is the sensitivity, can we see this reflected neurally? So we focus on two overlapping circuits, a cortical striatal circuit between the ventral striatum and the prefrontal cortex, and that ventral striatal palatal circuit that I mentioned earlier. So prior work has shown that a network of brain regions track subjective value of reports. That includes like the ventral prefrontal cortex and posterior cingulate cortex. And so we wanted to know, if the ventral striatum D2 receptors are shaping sensitivity to value, then perhaps maybe having more of those D2 receptor levels will be associated with greater modulation of the value signals in the regions of that circuit, including, for example, the ventral prefrontal cortex and the midbrain. So here we have a sample of 19 healthy adults who came into our lab, and they completed a delayed discounting task as before in one visit, and then on a separate visit had a PET scan with the radio tracer that labeled the D2 receptors. And for each subject, we construct a parametric map that represents the trial-to-trial changes in subjective value to an option, or the parametric effect of subjective value on the bolts. And to estimate the subjective value, we fit people's choices to this SA model, which is what I was just talking about earlier. So we estimate a K parameter, which is their discount rate, and can figure out how much people subjectively value two options that I have. So on the top left, what I'm showing you here is the average subjective value parametric effect across our participants. And it lines up with what we've seen in past studies, medial prefrontal cortex, posterior cingulate cortex. So nothing surprising there. And then what I'm showing you here is the average receptor availability or the binding potential of our tracer in the striatum with just a mask there on it. And then here are the masks for our regions of interest. And what we first observed here is a correlation between ventral striatal D2 receptor availability on the x-axis, and on the y-axis is the subjective value parameter estimate in the BMPFC. And what we find there is a positive correlation for both the BMPFC and in the midbrain. So we do find some evidence here that there might be a specific relationship between this modulation of the bold signal by subjective value in these regions. Importantly, when we just look at the unmodulated signal, there's no relationship. So this is really specific to bold signal that's been modulated by the person's own subjective value at the trial. So these results start to highlight a specific relationship between dopamine and motivation and highlight a key role in the underlying values. So now we add on to our findings. We find some sensitivity to the underlying subjective value of discounted rewards that might be modulated by dopamine. I'm not going to get the time to talk about this now, but feel free to ask me later about it. But we're trying to extend that lab study into people's day-to-day lives using experience sampling. And we're starting to see that dopamine receptors measured in PET seem to correspond to people's choices out in the real world when we ask them about different kinds of desires that they value. Okay, so now I'm going to shift over and talk about our extensions into the social domain. So in those last two studies, I showed you how dopamine supports motivation for monetary rewards and sensitivity to subjective value. But here, we wanted to understand these mechanisms for social reward motivation. So by investigating these social rewards, we can also evaluate the primary reward type in humans that isn't conditioned the way that monetary rewards are. So this is a better comparison to the rat studies that I talked about earlier. So everyone in the room, I want you to think about someone who is meaningful in your life, someone who you wish you could see more often. And can you recall how you felt when the COVID pandemic started and you knew you couldn't see them in person? How long were you willing to wait to see them? A day, a month, maybe 6 months? Well, your willingness to wait speaks to how much you value them and you value this social interaction. So perhaps like monetary rewards, these might be related to your brain's dopamine system. Recent studies have pointed us to the rewarding effects of social contact. In one study, being socially starved of contact was related to midbrain fold activation. And in some other works, dopamine in the amygdala have been associated with maternal bonding. Similarly, traits like social attachment correlate with lower levels of dopamine receptor levels. So given these sorts of connections, we want to know whether or not dopamine might bias how people differentially subjectively value social compared to monetary rewards. So here, participants who underwent a PET scan in one visit completed a delayed discounting task for monetary rewards and social rewards on a separate visit. So for example, they made choices about how long they'd be willing to wait to spend some number of minutes with a loved one who they indicated for us first. So they told us someone who they wish they could spend more time with, and they indicated how motivated they are to spend time. And so as before, we fit participants' choices to a hyperbolic discounted value model and extracted the subjective value estimates. And then we can test whether or not the preferences for the delayed option were associated with people's D2 receptor levels. So our measure of interest is the steepness of a slope in a logistic regression, and I'll to you now. And the idea is that if people have a steeper slope, they're more sensitive to that subjective value than they had a less steep slope. So what do we observe? So on the x-axis here, we've grouped our people according to how much D2 receptor levels they have into low, average, and high levels. And on the y-axis is the regression coefficient for this slope. So higher values means that the slope was steep. And so for monetary rewards, we see an increase, a linear increase across people who have low, average, and high levels of D2 receptor. So people who have higher levels of D2 receptors are more strongly weighed. They more strongly weigh this subjective value difference in their choices compared to people who have lower D2 receptors. And then when we look at the social reward, we see an interesting pattern emerge. It looks like, at least for people who have higher D2 receptor levels, there really isn't too much of a difference in how they weigh the subjective value between monetary and social rewards. But for people who have lower D2 receptor levels, that's where the differences really emerge. People who have lower D2 receptor levels seem to be more motivated by the social rewards than they are for the monetary rewards. And so potentially, social contact might be like a promising way to naturally enhance motivation for people who we know have lower D2 receptor levels. Think back to, for example, those clinical groups with different alcohol use disorder. So this is some evidence here that perhaps studying primary work in social tell us a little bit more about people's natural dopamine neurotransmission. So we're starting to see that specifically low D2 receptor levels are associated with higher sensitivity to the subjective value of social rewards compared to monetary rewards. And so I just showed you some work relating dopamine to the way that people value others, but I want you to now think about their distributions. And around the world, people face all kinds of challenges that result in unequal distributions of resources from healthcare to wages and education. And the decisions about who gets what are often made by just a few people in power with a few people in power in society. And so to explore how this problem arises, we study people at individual level. And we take a neural approach to understand how these sorts of inequalities might be exacerbated. An important concept to the study of inequality is how hierarchically structured a society is. So there's some work with respect to the brain showing that dopamine in primates depends on their social organization. So dominant monkeys that are individually housed and then move to a socially housed unit increase their dopamine receptor level expression. But we don't see that sort of change for subordinate monkeys. Similarly, when monkeys are given sulfuride, which blocks D2 receptors, you actually change the dominance behavior of those macaques. And then CD1 mice, which are naturally aggressive in a tube test, where they're basically just like a game of chicken, those behaviors depend on their D2 receptors. So we see that at least in other species, there's a strong connection between the hierarchical relationship between individuals. And at least from the fMRI literature, there's a network of regions that have been proposed to really underlie people's computations and evaluations of others in hierarchical environments. Some of these regions include like the amygdala and ventral striatum. And so here we wanted to understand in humans further, looking with pet settings. For example, past work has shown that social status is related to D2 receptors just on an individual difference level, and submissiveness traits. And when given tolcapone, which increases dopamine in humans, preferences for equal payoffs in an economic task increase. So there's lots of evidence for us to speculate that perhaps D2 receptor levels might bias people's preferences for fair distributions of money when in positions of power. So we're going to focus on people's D2 receptor levels in the ventral striatum and the amygdala. And so to study this, we had participants play a modified version of the dictator. So over several rounds, participants were endowed with a set of tokens that they were instructed to distribute between themselves and an anonymous partner. And these tokens on some trials were worth more in dollars for themselves and less for their partner. And then on other trials, those tokens were worth more for their partner and less for themselves. So we consider these to be advantageous and then respectively disadvantageous trials. These disadvantageous trials allow us to manipulate the cost of giving. So this is what it looked like for participants. They also played trials where the tokens were worth equal amounts between themselves and their partner, and they could just move these levers up and down. They can divide these tokens. So they did 20 different trials exchanges with 2 different anonymous partners. Okay, so what did we see? What I'm first showing you here on the x-axis, I'm plotting the amygdala D2 receptor levels, and on the y-axis, I'm showing the proportion of the total amount of the money shared with the anonymous partner. So the data is going to be split according to whether or not the participants were distributing in an advantageous or a disadvantageous condition, which favored better outcomes. And so the advantageous condition favors the participant, the disadvantageous condition favors. So firstly, what we see is that in trials with advantageous inequity, which favors the participant in yellow here, we see that participants with low D2 receptor levels shared more money with and that shared more, and that people with higher levels kept more money. This is in contrast to the disadvantageous conditions, where it really seems invariant to people's D2 receptors. So the amygdala dopamine receptor levels really seem to be sensitive to this advantageous condition. So next we'll look at the ventral striatum. And so here what we see is that participants in the advantageous condition and participants in the disadvantageous condition seem to vary with D2 receptor levels. So in trials with advantageous inequity, which favors a participant, people with more D2 receptor levels kept more relative compared to people with low D2 receptor levels. And people in the disadvantageous conditions shared more money when they have more receptors and kept more money with participants who have lower D2 receptors. So what we're seeing is that it looks like participants with high D2 receptor levels are just distributing inequitably generally. It doesn't matter if it's up to their advantage or to another person's advantage. They're just exacerbating inequity generally. And so what we wanted to figure out is, like, is this something that's systematic? Can it be explained by some other kind of rational utility model, given what we know about dopamine's role in facilitating the acquisition of rewards? Could it be explained by some kind of rule that's happening? Are people just maximizing rewards? And so by rational models, I'm just asking whether or not there's something systematic here. So to test this, we evaluated whether people are consistent in their preferences. What we found is that when we, for example, look at people's generalized axioms of revealed preferences, which is just the consistency of their choices, that does not seem to be related to D2 receptors. And then similarly, when we try to classify people's choices according to different sorts of moral prototypes, that also does not seem to be related to D2 receptors. So it can't really be explained by these classic models of rational action. And it seems to be something else. So what we find is that people who have more of these amygdala D2 receptors are less generous when they're in positions of advantageous power, and that people who have more D2 receptors in the ventral striatum are just exacerbating inequitably more generous. So we have some follow-up work now that a graduate student lab is trying to fit some models to, and trying to figure out if we can decompose this using models that quantify people's amount of guilt aversion and envy. Just to give you a sneak peek of some of the other directions we're looking at, we're trying to understand how social dominance more generally might be related to these decisions. So in an independent sample of participants completed this task online, we do see some associations with social dominance orientation, which just reflects people's preferences for social hierarchy. And specifically, people who have high social dominance orientation are distributing less fairly, specifically when they're in an advantageous position of power. So there might be other personality factors that we're going to consider as well. We also find similar effects when we ask people about their place of power in the workplace. There's lots of other factors that we're trying to tease. Just to sort of wrap up and kind of tie some things together, I showed some evidence in support of an idea that receptor signaling pathways may interact with baseline characteristics to influence people's valuation and motivation, and that these individual differences are important to consider. So while baseline differences could be due to factors like dopamine synthesis, it could also be due to other neurotransmitters like norepinephrine or serotonin, people's clinical status and medications. There's a lot of other factors that we need to consider when looking at these sorts of brain behavior correlations. I also showed you some evidence that suggests dopamine connects values to choices. Specifically, it might have a role in amplifying the neural sensitivity to subjective value. And by extension to that, we also showed that the amplification of the subjective value of social rewards might be particularly important for consideration of people who have low D2 receptor levels. So our lab is really interested in testing whether or not dopamine is more sensitive to specific aspects of social interaction. So for some individuals, older adults, for example, might have reduced dopamine function, but they remain highly motivated to spend time with others. They're so motivated for social rewards. Could social contact provide some sort of rescue place of lost dopamine signaling in a unique way? And then I presented some work that indicated a role for dopamine in costly giving. specifically when people are in positions of power. So this role doesn't appear to be driven by some other rational utility model. So some next steps in our lab include understanding the basic mechanisms that might explain why dopamine might have some contribution and some individual differences. So in the remaining time, I'll just touch on some ongoing projects and some future directions. So these are asking questions about how people are integrating social information into value-based choices, how motivation supports decisions in daily life, and what is the role that dopamine plays in social decisioning more broadly. So with some collaborators at UPenn, for example, we've been working to understand how people use stereotypes in their decisions about others, and how do multivariate neural representations of these stereotypes get encoded when making intertemporal decisions on behalf of others. So in this experiment, people are making delayed discounting choices on behalf of people who have different names that vary in stereotypes that are associated with warmth and competence. And so while we know people have these stereotypes in mind, how are they actually weighed and valued when people are making choices? And so what we find, for example, so far in our preliminary results is that the ventral medial prefrontal cortex seems to be representing this dimension of stereotypes of warmth and competence when people are making these choices. Secondary, we're interested in understanding the underlying mechanisms that explain how people learn and unlearn impressions about trustworthiness. So specifically, we're using a paradigm in which participants will interact with different targets who are viewed as trustworthy or untrustworthy. And they have to learn through experience whether or not the sort of trustworthy appearing faces are actually behaving in that congruent way, or are they behaving incongruent? So maybe you see this person, they look really untrustworthy, try not to share money with them, you know, and then they decide not to share money back. So this is just one example of how we can sort of enhance the richness of social information to these more basic studies, which I showed you earlier, where people were just playing with anonymous partners. And in another vein, we're trying to understand how observing other people's behavior during reward discounting might guide subsequent decisions. So specifically, we're testing whether observing one's preferences and expenditures for high effort versus low effort options impacts cooperative behavior. So for example, you see this person, they are just choosing the easy way, and you see that they didn't really work hard for that reward with button presses. Later on, when you interact with them, do you decide to share money with them? Do you think that they've earned it? There's lots of great work on social psychological literature that suggests that people have these sorts of moralized opinions and ideas about effort and value, but this hasn't really been tested experimentally yet. This will allow us to really understand what are the computations that are underlying this moralization of effort. And then in a more exciting domain for us to connect to dopamine, we are trying to take advantage of a new simultaneous fMRI PET scanner to understand how social rewards might evoke endogenous dopamine release, and how, for example, when people are playing with computerized agents versus actual human beings, does on the at least minute level, can we actually see fluctuations in people's dopamine levels that suggests that people are more sensitive to this social context in the same decision, in the same kind of decision. So we'll be doing this with an ultimatum. This is more long-term. Be happy to give you some updates once we start our data collection. And so I want to thank my plenty of mentors and collaborators who've been instrumental in the work that I presented here today. So thanks to all the participants in our studies who spend time with our molecular imaging and safety protocols. And I want to thank funders with whom this project would not be possible, especially this DSPAN award, which was funded by the National Institute of Neurological Disorders and Stroke. These kinds of awards are really essential to this kind of study and the kind of work that allowed me to get to where I am now. So if there are any graduate trainees here who are interested in those kinds of funding mechanisms, please talk to me. I know that it's a more challenging time now to get these kinds of rewards because they're not as available, but there are always clever ways to find money everywhere. So thank you all for your time and attention as well, and I'm happy to take any questions you have.
I want to ask a kind of basic methodological question, which you kind of alluded to in your last slide with the simultaneous fMRI, which is, if I understand correctly, measuring the receptor availability is basically putting an upper limit of the impact that dopamine release can have on the brain system that contains those receptors. It doesn't tell you anything directly, does it? An individual, if you're doing resting state PET, it doesn't tell you anything about the receptor occupancy. So in other words, you could have two people with exactly the same receptor availability, but when you put them into your experimental tasks, one of them releases a lot of dopamine, the other one releases very little. From your metric of receptor availability, they would be considered to be equally sensitive to dopamine, but behavior different, different occupancy and stimulation of those receptors. So is that something you worry about in those early studies that you're describing?
It's a good question. So I would say not in our samples. And that's because from work where people have scanned people using these D2 receptor ligands to visualize the receptors, and then in those same people visualize the dopamine synthesis capacity they have presynaptic to different treatments like F-DOPA or FMT. Those are highly correlated with each other within individuals, at least in healthy people. So we're not too concerned that these things are going to be like that. You know, if the synthesis capacity is going hand in hand with people's receptor levels, we're not really going to see that effect of not being able to capture something else that's dynamic there that might be explaining the differences. But if people, for example, vary in their endogenous levels, for example, because of psychopathology or something else, that's where I think those differences would emerge. So it's definitely something there. It's another thing we're not measuring.
Just to quickly follow up, what is the time course over which dopamine receptor concentrations there? And maybe they changed.
Yes, so it depends, I think, on how we're thinking about dopamine. Are we thinking about like these impulses that are firing at the sub-second level, which we can observe in rats, can't think in humans. So there's some work that's starting to do this in patients with epilepsy, where you can use fast scans with telemetry, and the time is pretty conserved there. Some work has shown that you can observe the sort of transient changes down to about 3 to 5 minutes, and that can be visualized with some of these tracers.
Thank you.
Yes. Yes.
Thank you for amazing. I thought the social version of related content was very creative and interesting, but at the same time, I thought that it would have been difficult to make this monetary and social task compatible. And like the value of $20 not the same as the value of 20 minutes. I was curious about there was any procedure that needed to make these two tasks compatible.
So I have two parts to that. One is that we did it, but what we're doing now is actually we have a graduate student who's trying to analyze that data and see if we can estimate some sort of willingness to pay. So because participants did both of those tasks, we can and the time delays are the same, we can, based off of their choices, sort of assess whether or not there's some equality or estimability of the dollar amount that corresponds to like the amount of time for the person. So we are trying to make this task better, but this is like sort of like a first pass of creating a task way back. And at that time, we didn't make that consideration. I will say though that despite that, we do see individual differences across, for example, in age. So in that same task, we found our teams found that people who are older have a preference for spending more time with the person. And we don't see that sort of age-related effect in those same participants with monetary rewards. So it doesn't reduce our ability to see like individual differences that still emerge.
Maybe I'll ask something we talked before a little bit, but just like a general thought about how that type of research that you're doing, say like looking at trustworthiness, can apply broadly and think about social media and how do people decide what to do and they interact with other people and those are other agents. Like so, just broadly thinking about relevance of like really interesting and very fundamental basic research in a more societal level.
Yeah, so we really think about this in terms of, we talked about trying to increase complexity, increasing the social complexity of these decisions. So in the way that we've designed it, there's some immediate connections to the way we understand susceptibility to fraud. That's something that people are trying to resolve ways to reduce susceptibility to fraud, victimhood, and how people are trusting others, whether it's a financial scam or something somebody tells you to, like a phishing scam or something like that. Those are some like the more immediate connections where there's limited information that people are relying on to drive their choices. But you bring like a really interesting point that trust in today's digital age, there's a lot more complex information that people are trying to resolve, and they might be trying to integrate information with their pre-existing biases. I didn't get to talk about that here, but some of that work what we're trying to do is connect people's initial biases about others to what they have to learn over time. So, for example, if you have some sort of pre-existing bias about the way someone looks, or the kind of occupation that they have, or the kind of name that they have, how easy is it for you to learn or unlearn through repeated experience with that person, whether or not you should trust them? Some people might be really quick to unlearn that and break that association, and some people might be slower. Identifying that sort of speed, or that which people take to unlearn or break off their behavior, I think is going to be really important for us to identify what are the ways that we can reduce the sort of issues with trust that we see with, for example, social media and trusting some video you saw on TikTok or Instagram because it conformed with a belief that you already have. So how do we kind of shake that initial? And that's like our long-term action.
So I was trying to think about how to ask this question. A lot of times when people talk about social decisions, I think two things get conflated. They talk about economic decisions that have a social component versus decisions that are social in nature, where they're actually about like spending time with someone or something like that. So I'm curious, like here, it sounds like you're doing a economic task that's going to have a social component to it. Do you think that you may have a different result than you would if the decision was actually social in nature?
That's a great question. So I'm trying to think about the kinds of decisions that you're thinking about social nature. So in this case, we're thinking about an ultimatum game with human partners versus non-partners. And
thinking of the reward as actually as a social reward as opposed to a secondary reward, which is an economic reward.
That's tricky because I think that for dopamine might have some kind of double duty here. It might be amplifying the reward itself. It's a primary reward. Just spending time with people, knowing that it's a human is enough to elicit some sort of response. And then the actual reward that you get through an exchange, like adding onto it. So one way that we could probably tease this apart is changing the way we incentivize people in an experiment. The way that people can get bonuses or not might vary. And that might be one way to subtract the rewards that they actually get in social context and still keep like a social person or actually actualize it by having people come in with friends. That's a great question. But I do think that dopamine is affecting both. I think it's amplifying the social reward, but also there's this context.
Are there any questions online? Disembodied voice.
As of now, there's no questions.